I think
I
it's
love
just all Wonder Woman.
Yeah, Star Wars was just insane. I thought that Star Wars was Star Wars. Apparently not.
Yeah, it's really good.
George Lucas just like, this is pretty cool.
Because you know what that was like.
Make the silly kids.
You did not have to add that.
I know.
It was just like, you know,
That's actually the one thing I did. Like I knew.
and still.
It's kind of dense.
The paper.
Yeah. I
I
finished
it. I just like talked that out as a kid. Like I watched it. And I was like, you get a on we looked at each other like, what? Oh. I see this for like 2. It's writing it. But it's not a problem. It's not though. Get to go to bed now. You're like. I don't think so. I'll check the updates. What was it, the 26? If I'm. Oh no, you have to look at the stuff. Look at the class, like, that's a.
No.
Oh no. So, I'll make it. I'm emailing them right now.
You're actually.
So basically, no, it's not.
So. Study more.
You have like one in front of you, one behind.
I'm so mad. Every time I visit, I can't like hear good things.
Split, and this keeps like.
All right, everybody, welcome back. It was a good break. All right, everybody have fun? Yeah? Relax a little bit? All right, cool. Let me finish. For the midterm, the bright space part. I think the average was. Yeah. Yeah, so, good job. The other thing I did notice is like when you guys took the quiz, there was big bumps. Like from the first time you took it to the second time, so doing it twice helped you guys out. One thing I forgot to make it really clear was you gotta be very careful with the wording. Make sure you're reading like very precisely the words that I'm writing down. Like I know the more sort of genotyping, I got a lot of you guys with it. Like when I was talking about heterozygous or homozygous, or if the parent was gonna be wild type, or if the one is genotype or genotype. I still gotta finish grading the second part, the written portion. The numbers you guys do a lot better in that section. I don't like the multiple choice stuff just because it's like either you get it right or you get it wrong. Even if you know some of it, you don't get anything right. So no, that's what I really like about like our open-ended quizzes. So if you think about it more this next week or so, and then we'll talk about we can plan the next midterm. Or so then you guys still want to go in class, or if we do this like online bright space and half written portion, we can do that half written portion in class. Yeah. I'll see how we'll balance it out. But let me finish grading and then we can have a good conversation about that. Let's see what else. time wise, I think you guys did all pretty good, right? I gave you guys 100 minutes. That was pretty good. Yeah, it was a lot of questions, but it was good. It was good. What else did I talk about? Oh yeah, you guys have your papers I did today. I'll open up. Thank you for reminding me. I'll open up the Brightspace submission thing, so you guys can upload a PDF copy, and I'll grade it. I'll give you the grade there. But remember that I prefer you guys to upload a Word document and then share with me on Google Drive. And that way, I'll give you all the feedback and comments on that paper directly. It's a lot easier for me to work with that one. So we'll try to get those to me by the end of today. I'll put the deadline to be midnight, but if you need extension, just let me know. I'm not pretty great. But I think I've never caught up. And then what else? I think that's it. There we go. All right, let's go. So today let's talk about the plasma membrane. So we're gonna talk a little bit about like this, how it works and kind of how it all comes together. It's really like gives us this really powerful control inside of the cell. So let's get into it. So for this, you're gonna see that basically the plasma membrane is gonna help it to interact with the world. At first, it acts like a barrier, right? It protects us from interacting with things, but at the same time, through it, we can actually set up these really nice controlled ways of communicating with our neighboring cells or even with the environment on itself. So getting a little bit deeper, you're gonna see we're gonna kind of go through how the signals can go from going outside into the cell without the molecules actually coming in. All right, so let's see how the plasma membrane works. So for here, I'm showing you guys an electron micrograph, right? So here we're taking a picture, and for this one, if we Photoshop it, we get something that looks a little bit like this, right? So we're gonna be focusing on is gonna be right here. This orange section is gonna be outside the cell. This blue section is gonna be inside the cell, and here's where we have our plasma membrane, right? So within that, the big key features you guys have to be super comfortable with, the vocabulary is gonna be the different groups that we have there, which are we have hydrophilic regions and hydrophobic regions. Right, so hydrophilic, what does that mean? Hydrophobic? Perfect. In my bio 133, I remember I used to pick on my kids with the amino acids. I'm like, I need to memorize them all in this way. But the big thing about it is because you have these two different groups, in each side you're going to have the lipid bilayer. So you're going to have two different molecules setting up. So when we're looking at it, we're going to have our hydrophilic side. It'll have its tail, hydrophobic side. This, let's say, would be like facing the outside. We'll have like an image, like a mirror image of it on the inside of the cell, and then hydrophilic and its hydrophobic tails, right? All these molecules are kind of just sitting right next to each other, kind of like I almost imagine like if you guys, like the whole class with the plasma membrane, each one of you would be one of these molecules, right? And we'll say your head is your polar side, your body is your hydrophobic side, right? You guys are kind of set up in this like 2D structure, but technically there'd be another like image, like a mirror image of you guys right below you, and they're going to be sitting the opposite way. When a molecule is trying to get through this thing, it makes a really cool barrier because on the outside, these things are able to interact with the water, right? So if you have ions like water, water that have like salt, so that's sodium chloride, they'll either have a charge, which makes them polar, or they'll have like they'll try to do that hydrogen bonding, right? Like we do with our water molecule because the flow is negative. They're happy interacting with the red side. They can't interact with the yellow side, right? So if that molecule's out here trying to bounce around, it might be able to interact with this part here. The moment it gets to this like hydrophobic region, your water molecule doesn't want to interact with it. It's more favorable to interact with water than to interact with the wall. At which point it basically acts like a shield to these ions. So at that point, it lets us control water inside and outside of the cell. Not control, isolate. We have water inside the cell, we have water outside the cell. At the same time, you can have a difference in ions. You can have more ions inside or outside the cell because they can't actually get through it, right? So like salt can't just go right through your plasma membrane. It also can't just go from the environment into your cell. It has to find a way of doing it. So really cool feature. Make sure you guys feel comfortable with it. And then we're gonna really dive into basically how these guys work or how they get their job done. We have time. We'll watch the video at the end. We'll come back. All right, so now, like you said, we had that huge wall made up, outside, inside the cell. And now we're going to zoom into one of these and we're gonna look at the structural features of them, right? So the big thing that I want you guys to feel comfortable with here is, I call them like the body and the head, that's how I remember them. So this should basically be the body. So there's a little glycerol sitting kind of here, these 3 carbons. But then the big thing is these orange sections, right? Which is basically a carbon chain, right? If you guys remember your old school chemistry drawings, when they draw them like this, and it's a single line, we're saying that there's a single bond happening between each carbon, right? In order for these carbons to be happy, they need to have 4 partners. This one has 2 carbons. It also has 2 hydrogens like this, right? So this one would have its 2 hydrogens. And it keeps going with the carbon chain, 2 hydrogens, 2 hydrogens. And this one binds up with that double bonded L. The carbon, right? So here you can see single bonds, you have 2 hydrogens. This is what they normally call saturated. Because carbon and hydrogen have like the same electronegative charge, then basically this will really make them like non-polar, it also makes them hydrophobic. So each one of these places, like each carbon, I'm almost thinking in my head like it's part of that body of places where you don't want to interact with water. The length of the tail, like the longer it is, it means it's a longer region for your water molecule. If you want it to come into the top, like to the top side, you would have to try to get through all these interactions to be able to break it. So that's really the big thing that's kind of helped protecting that. So you can see that's pretty similar on all of them. You're only going to see one big difference. I'm going to talk a little bit more later, but it's the unsaturated, which means we're talking about how many hydrogens they have. Sometimes you'll get stuff where it looks like this, but then they'll have a double bond, like that. Because it has a double bond between 2 carbons, now this already has 3 partners. It only has 1 hydrogen, like this. The thing about that we're going to care about is when you have saturated, you're going to have this perfectly straight lines of the carbon chains being built in a straight line. When it's unsaturated and there's double bonds, you have less hydrogens. This thing is going to take a new angle. So instead of being divided in this way, where they kind of do like that pattern looks a little bit like this, right? Like the tetrahedral. They'll shift over to like they'll be on the same plane, and they'll be divided into 180. No, not 180. But they'll divide it basically into 360 will divide it into 3 angles, right? At which point, normally this thing is going to make a kink out on one of the sides. The issue with that is be like, right now, all of you guys in the class are kind of sitting like this, saturated, right? We can all sit right next to each other because we're all sitting straight. But if one of you went and stuck out your arms to the sides, you start pushing each other, that would end up making holes into the environment. So that's where I'm gonna talk a little bit later about this is gonna really mess us up. But that's on the tail side, right? Then on the top side, on the heads, like, you know, thinking of the head, you can see you can have different groups that are gonna be attached. So some of them, we're gonna call them phospholipids, right? So phospho, there you're gonna see in that dark orange, there's your phosphate group, right? So your P with your 4 oxygens. On top of that, you can have these different modifiers, right? So here you can see it's inositol, right? There's some, this is a glycolipid because there's a sugar ring. Here you see more carbons, a little bit of nitrogen. Here's your sphingomyelin and phosphatidylserine. In the book, they really go over which ones are enriched on which side of the membrane. That's one of my favorite quiz questions. And I like the way that they already connected it to you guys, that it's like when you go from the ER to the plasma membrane, the inside of the ER ends up on the outside of the cell. The outside of the ER ends up on the inside of the cell. Yeah, so make sure you guys write a table about where these are located, where you find them in the ER, and where you find them in the last part of the cell. All right, so these are it's one of the big groups. The other big one you're going to be looking at is going to be cholesterol. So this one has a little bit of a different shape. So for this, it's like, this is our polar groups, right? Or like the charge heads. There's our negative charge. There you see different charges. For cholesterol, it's that, right? So it has a really small charge, but if you imagine this to be the body, it still has a pretty big body. You see a bunch of carbons with hydrogens. that means your charge is kind of like it's hydrophobic interaction. Right? Later, we're gonna talk about how you start messing with these, when you put them into the membrane, these would like fit nicely packed into each other. And then here comes in your cholesterol with like this thing. Right? So you can see it has a little bit of a charged area and it has a big body. Again, we're gonna play with the dynamics of our membrane. All right, so let's go to the next slide. The other big component of the membrane is gonna be, you guys see we're gonna find a lot of proteins that are gonna be found on the membrane itself. These are gonna be the big players. One of the big key functions that I really like that they do is that they can move molecules around. So, like I said before, plasma membrane is huge hydrophobic region. It's like a little shell where we can't get water through it, we can't get ions through it. That's where some of these come in. Those are my favorites, but they'll be like proteins that actually go through the whole membrane. So the opening, have an opening on one side, go completely through the structure and connect it to the other side. So these ones are called the transmembrane proteins, right? Because they go across the membrane. You can also call them integral because they're inside of the membrane itself, right? So for those, the ones I really like are water transporters. I'll probably bring them up a little bit later, and ion transporters. So those are basically, like I said, the ion can't make it through this section. This thing ends up having these alpha helixes, which are amino acids that have certain regions that when they stick out, they'll be hydrophobic for a stretch of that alpha helix. What that allows them to do is they can sit inside the membrane, interact with the hydrophobic regions, and it's technically hydrophobic with hydrophobic interaction. It'll mix in right. They'll set up like these outer parts, like multiple alpha helixes. On the outside, each one of them will be hydrophobic, so it'll sit in the membrane and it'll be happy. On the inside of the alpha helix, they basically make sometimes they make a channel, so it'll be like a big pore, and the molecules can go in and out. Some of them, they have like ATP synthase, or another one of my favorite pumps. It literally has pieces of the amino acids that kind of rotate, and they can move the ions against the gradient. Like that's how powerful these guys are. All right. So when we look at the proteins, if you go weight, if you get cells, pop them open, divide lipids and proteins, what you get from the plasma membrane is 50% lipids, you get almost 50% proteins. So it's a lot of proteins that are going to be present. The other thing we're going to find with proteins is you're also going to have some of these called peripheral proteins. So these, they basically interact with proteins that are bound into the integral proteins, or they can bind with more peripheral proteins. But they themselves will actually go into this membrane or cross the membrane. That's going to be really important later on when I talk about cell signaling. Because normally what we have is sometimes we'll have something like this. It's going to be the receptor side. Let's say a signal from another cell comes, it binds here. This modifies the conformation. The signal gets transmitted. It modifies the interaction here. In some cases, it'll release the red protein and then it can function. So it's super cool mechanisms. Receptors, yeah. So we're going to get into that for this next section. All right. So back to thinking about how that membrane works as a whole. So there's different types of movement that we're going to see within these membranes. So in that case, here what we're really talking about is how the membrane moves, but we're talking about the same plane, right? So we're only looking at one side on the outside, and we want to see how it moves around. So that would be my analogy to the classroom, would be like if you guys in your little rolly desk started moving around the classroom, the membrane can move around like that. So you can see here, they brought in cells from humans. They had a surface protein that got bound by an antibody, and then this brought the red fluorescent dye, right? For my cells, again, they got another cell, they used the surface protein, they tagged it with an antibody, and they made them green. When they combined them, after 40 minutes, they saw something like this, right? So it means that plasma membrane, again, on this side, was shifting around like this. So if we're doing the class analogy, you guys will basically move around your desks, right, when we're doing the group works. So they can move in this direction. There is some parts that are constricted. For the most part, this kind of movement is easy, right? Hydrophobic regions interact with hydrophobic regions, and your polar regions interact with polar regions. Everything's going to be good. Put it in here or no? Where did I put the slide? All right, sorry. So I'm missing a slide, I'm missing one of the slides. There's a second kind of movement. I'm gonna go into a little bit later on for the signaling part, and it's when it's movement from the different sides of the sheet, right? So in some cases, like I said, there are molecules that are enriched on the outer part. Some molecules are enriched in the inner part. This movement, these phospholipids can't do it by themselves, right? If you try to grab this molecule, let's say the phospholipid came in, and you try to flip it, that charged head would have to go through all those hydrophobic regions, right? And that, again, doesn't want to do that interaction, so it wouldn't let it flip. So in this case of like, we have a lipid flip from this side, flip it over to this side, it's not gonna happen on its own, right? There are some proteins that control it, and I'll talk, like I said, I'll talk a little bit later about them when we do the signaling aspect, but this movement doesn't happen naturally. We can force it with another group of proteins. So pretty comfortable with the movement? Yeah? All right. So now let's look at the transport of it, how we're gonna get them getting made and how they basically move around. So here, let's go to, so we'll start at the ER, right? So remember, there was two types of ER, do you guys remember? Smooth and rough, right? So rough, what does that give us? Proteins, right? Ribosomes make it rough, we get the proteins, and then the smooth side, lipids. There we go. Yeah. So that's what gives the lipids. So we're gonna start getting lipids right here. Inside the ER, you guys remember when we talked about the protein degradation or the protein folding? We have glycosylation happening. Like we're adding sugars to things. Same thing is happening here to the phospholipids that we make. We're making them on the inside of this membrane, but on the inside, they're getting modifications built upon it. This thing's gonna form a vesicle. That vesicle is basically gonna move from your ER. It's gonna move into the Golgi, right? So we do a little. My cartoon version of this. So in this case, let's say this is my ER, and this is where we're starting to blood off. Right? If we zoomed into one of these, we'd be able to see a phospholipid bilayer, right? So in that case, we have the inner wall going like this, and we have the outer wall. Right? So this vesicle basically is going to start protruding. It's going to get this shape. Eventually, this thing's going to kind of go back onto itself. It'll collapse. It'll look a little bit like this. Right? It'll start pulling itself tighter and tighter. Eventually, this will pinch off. So this will close up here, and this will become a little small vesicle bubble. Right? In that case, again, the inside, where we did all the sugar modifications, are going to be like that, and we'll have it outside of the cell, just like that. All right. We're going to send this out, and then we're going to go bump into our Golgi, right? I'm going to draw just one Golgi just to make it easy. This would be my Golgi. This vesicle, you imagine it, it's moving. It's going to get into this Golgi. Now it's going to crash into it, right? When it's doing that, it's going to be brought together. There'll be like proteins that pressure these two things to become together. So this bubble, right, will bump into it just like that. It'll push so hard, it'll connect itself, and it'll give us that structure, and eventually it just becomes a part of it. Now, when it's doing that, thinking about what we had here, when we redraw this, remember we had the outer phospholipid part, the outer layer, and we had our inner part like this. So the inside of the ER, which would have been that green side, goes into the inside of the vesicle. When the vesicle gets over there, it dumps into the next compartment. It now becomes the inside of the Golgi itself, right? Kind of how we saw this experiment with the human and mice cells, that the moment these two membranes collapse into each other and they become one, they'll mix into each other just like this, right? This exact same thing is going to happen the moment we send our first vesicle into the Golgi. This thing's going to go into itself, all the material that you have here is gonna start spreading like that onto the Golgi itself. Right? So the material is gonna go diffuse into the membrane. It goes through the Golgi. Just I'm not gonna draw little stuff, but we get to the other side. So imagine your stuff in the middle, right? And it's time for us to make a new vesicle, just like that. At which point, we still have the same thing. On the inside, we're still adding more modifications to our phospholipids, but the inside phospholipids will stay here. The outer phospholipid layer is gonna stay right there. Yeah. At this point, again, we pinch it off, we're gonna send it, and it's gonna go to the plasma membrane, which is what we have out here. Right. When it does so, it's gonna do the same type of behavior. The inside is gonna stay on the inside. Once it hits, it's gonna dump itself all the way up to the environment. So what ends up happening is, if we end up looking at the, let's imagine this was the barrier. I'll take a little. So here, we're about to exit the cell. This is your plasma membrane. When this vesicle comes and it's gonna connect into this, it's gonna do the same type of attachment, right? Except this time, the green side, it's gonna look like that. And then the red side's gonna be on the outside like that, right? And when it dumps its content, what we're gonna end up with, it's gonna be that everything that was on the inside of the ER, it's gonna end up on the extracellular side, right? And everything that was on the outside of the ER ends up into the cytoplasm. That make sense? All right, so like I said, we have the 5 phospholipids we talked about. They do 2 and 3. So I need you to figure out where they sit in the ER, where do you think they would be in the Golgi, and then where would they end up when they get to the plasma membrane? Yeah? All right. This is looking pretty good? All right. This one. I don't want that one today. All right, I'm gonna stay on the membrane side. I don't want to get into the proteins quite yet. So we're gonna talk a little bit about how it works in the sense of like its interaction with the proteins. For this one, let me think. All right, so I'll go to there. Let's go. So I'm gonna focus on the protein side. If you look at that one, again, we saw that the plasma membrane has that function, right? We have our hydrophobic region and our hydrophilic region. When you're trying to figure out how it works, you can try to break different interactions to try to pull out what's gonna happen, right? In this case now, they're basically focusing on the proteins and how they keep themselves onto this membrane. That first one, the high salt concentration, it's gonna be, if you imagine there's gonna be a protein, it's gonna be sitting in the membrane itself, and it's gonna be interacting with like another one of these peripheral proteins, right? The only thing holding those two together is gonna be protein-protein interactions. That normally means you have a hydrophobic bond, a polar charge, right? Something's positive, something's negative, that's holding them together. The moment you dissolve something like NaCl, you make an Na ion and a Cl ion. You have 2 different charges, they'll go and grab onto those proteins and they basically cancel out the interaction, right? So high salts, it'll help us get rid of all those proteins that are not integral. If it's not attached to the membrane, and then the salts, all the bonds are gonna break. So you can separate the proteins one way. The other really cool one is gonna be the sulfide linkages kind of change the shape of the cell, but this is gonna be really the big one down here, which is gonna be using strong detergents. So the cool thing about how detergents work is that we've messed with the dynamic of what like a phospholipid looks like. In this case, here's your hydrophilic group, right? It's small. And again, here's your hydrophobic tail. In this case, you can see we have about 6 carbons in the middle, right? So between these 2, we have about 8. If we go back to the other drawings, these, they have a huge range, right? So this is carbon 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So this tail of 15 is going to be a long tail. When you're bringing the soap, it's going to be about half the size, right? That change in size is gonna give you a soap that's gonna look something like this. There's your charge, but your body's gonna be short like that. Again, if these molecules are sitting and trying to interact and bump with each other, you basically made a huge gap down here, right? What's end up happening is if you make them even and they all line up, they're able to stay straight, right? Or they make they're not that good at bending. It'll be a flatter structure. You bring in those lipids, now there'll be like tall, and there'll be a short one. They can collapse into that space, right? As you collapse more and more and more, this is gonna end up producing like a bigger bend. It'll make your membrane fold into itself like that, right? Just because you have like that smaller base for each one. So when you're doing a soap or detergent, I think from my other class we did like Dawn soap with the chemicals. The shorter, like these short tails with this charge will basically disrupt this whole membrane. It'll make this thing want to fall apart, right? The other way that it attacks the proteins that are inside of that is by using this type of interaction, right? So they're using their hydrophobic tails to bind to that alpha helix. That alpha helix, which before was hidden in that hydrophobic region, now can get clustered around like hydrophobic pockets. But because they're antibiotic, now the hydrophilic part will be sticking out, right? Now that means that this is hydrophilic, and technically this on the surface is also hydrophilic. So that instead of having the protein being trapped inside of lipids, now it actually can be free and it'll float off in solution. So when you do the spinning, you try to separate things by size, you'll be able to pull them out. So this is the old school way of separating the two types of proteins, those that only sit on the surface versus those that are actually inside the membrane itself. But at the same time, this is a big, powerful molecule that you can use it to help dissolve the membrane into itself. So like, so like bacterial infections or viruses, they all have plasma membranes. That's why you just use soap and water, and you basically have the soap like go into the plasma membranes of all those bacteria. It'll make them, instead of being like a big cell, it'll start shrinking them up and make little smaller bubbles. So soap water will kill more stuff. What's the other one? And I think ethanol messes with it a little bit too, but soap and water works pretty well. All right. So talking about how that plasma membrane structure, it itself, this one, right, with equal length tails, they prefer, like if you just grab water, these phospholipids, and you mix them around, some of them will turn into sheets, some of them will turn into bubbles. If you put in more shorter molecules, they'll more likely turn to bubbles. So they're able to kind of give you a little bit of range. These pure themselves, they can give you a little bit of bend. When you add these, they'll bend a lot more. Right? So the thickness of this membrane, the length of them, is going to modify how it behaves. So it means that it itself has a bit of bend that it can accomplish, but normally it doesn't give us mechanical force to make it happen. So here, this is a really cool example. It's one of my favorite type of cells. So these would be your epithelial cells that are sitting on your the inside of your stomach. So in this case, they have this really cool thing called microvilli at the top. And basically, it's like, if you imagine this table was a cell, and this would be the side facing the inside where your food is. Here, they basically deform the plasma membrane to make like these little like finger-like projections, right? All those finger-like projections basically give you a crazy amount of surface area for you to be able to pull more molecules in. The plasma membrane then is able to create these really nice folds. Like here, you can see there's a lot of stress, and there you have a lot of stress. At this point, it's not just the chemicals that are found in those lipids, in the phospholipid bilayer, but there's actually proteins that are helping anchor these structures together, right? Let me find you a picture of that, and I'll put it onto the slide. But there's like a whole mechanism of anchoring these bottom sides of the phospholipids bilayer. You can make these nice structures, and you kind of poke them in from the bottom, so it kind of puffs up, like they roll up like balloons. The other place where you have this really cool stuff happening is going to be right here, where you have these tight junctions. So at which point, again, this is our stomach cell. On the inside, on this side would be where we have all the food. On the other side of this, where you have your blood vessels, right? You don't want your food, the acids, all that stuff getting from this side into your actual cells, into your bloodstream. You want it to go through a cell first. So at which point, it's like if this is a cell, there's another cell to it, you get proteins that grab onto each other. They produce from this cell, and they grab on so tight, they basically make it so nobody can be fused to this part of the cell. So it makes you like a little bit of a seal around this one, right? They do that with each other. So there's gonna be a lot of integral proteins, peripheral proteins, all of that, holding this domain very, very tight. Right? Like I think it's leaky gut disease is the one where people have issues with this protein, and then food sneaks into their bloodstream. They have issues because of it, like immune responses. But that thing is so tight, there's so many proteins holding on that as much as you're stopping the ions, it also kind of like grabs on so much of the phospholipid bilayer, that thing doesn't move around that much. So the lipids in that thing are kind of caught within all those structures, the diffusion gets slowed down. So yeah, so it's super flexible, but normally other stuff, like the proteins, are basically gonna give it like that big final shape. So part of that, the other thing we're gonna find with the shape of these phospholipid bilayers is there gonna be these little lipid rafts, or gonna be kind of regions that are gonna be different compositions. So in this case, we're looking at the ones in purple. And kind of here, zoom in, kind of looking at the little modifications that you see for the tails, right? So in this place right here with the orange section, you see we have the long tails, everything's next to each other, nice phospholipids, they're gonna be happy. In these lipid rafts, sometimes what you get is you get higher concentrations of shorter molecules, like this, and then they actually aggregate better with each other. When they do so, they change the width of this thing, of the phospholipid bilayer. There was a cool paper, I don't remember, people were published on it, but they were like, the shorter regions made it so that the width of the phospholipid collapsed a little, right, to kind of compensate for these shorter little ones. It got smaller. Within that smaller region, there were certain proteins that had smaller alpha helixes, right? So a long alpha helix will let you get across this whole thing. But if you have a shorter alpha helix, you might only be able to make it to a smaller section. So they were saying that the lipid rafts are holding all these type of components together from the lipids, and they were also holding together certain types of protein. The cool thing about that was that they were controlling the cell, they were looking into how the cell was controlling where it would send the specific fats, right? To be like, hey, we should send short lipids to this side, we'll send the proteins that can work with that. And like going back to like my favorite cells, the epithelial cells, that's how they were controlling this kind of stuff, right? Because on this side, you basically need pumps that grab sugar and they pump it into your cell, right? Because that's the whole goal. You're trying to digest food, and you bring sugars and salts in here. So then you would enrich sending the sugar transporters and ion transporters to the top of the cell. Those are made to go from molecules from outside the cell and you pump them in, right? On this bottom side, the goal here is this thing takes up the food, then you need to push this out into your environment, right? So here you have a different set of proteins working there that are helping push proteins out. So, sorry, sugar out. So sugar in proteins, sugar out proteins, right? If you send them to the wrong compartment, you'll starve to death even though you're eating, right? So it's super important you send them to the right spot. So yeah, I don't want to go heavy into this part. All right, I'm gonna adjust these slides. It's gonna be too heavy. I don't feel like it today. We just got back from break. But here, I'm gonna put an easier slide. I want to talk about temperature. And I'm gonna put one about cholesterol. All right. So one of them is, again, these sheets, the way that they move around, it's like you guys in the class. You can move on your desk and you can move like this, right? The hotter, the higher temperature that you have, all that really means is that the molecules are moving faster, right? So the stability of that membrane, the hotter you get it, they move faster and faster. It makes it harder for them to kind of hold it together. The molecules are bouncing around. On the other side of that is when they're compressed like this, like let's say we only have this type, and you have really low temperatures, these molecules aren't moving around that much. So what they're going to do is they're going to basically kind of settle into each other. So you're like, your desks will basically collapse into themselves. So that affects how you freeze. Having different sized particles, like the different sizes of tails, or having like the cholesterol with the funny tail, some of those things, what they do is they help make this thing not as stable. So when we get to really low temperatures, it's harder for them to freeze. So some animals use it, some plants use it to make them tougher. So I'm making a new slide for that second, it's easier. So that's really where the types of fat really affect how you behave with the temperature. Making you stable in hot regions, also it makes you not freeze at really, really low temperatures. Yeah. All right. Yeah.
So saturated doesn't freeze.
I think it's, let me look at a picture real quick. Saturated. I always make these two look, guys, I'm sorry, give me a second. Butter is high in saturated fats. Yeah. So butter would be sitting on this end, right? Long tails, but they're all perfectly straight. So it's very easy for them to clump up together and become a solid, aka butter, right? And then I think your unsaturated fats, they have these kinks into them. Because of the kinks, the things are trying to settle in, and there's one that's kind of maybe, it's like, it's pushing the other things on the side like that. So it's harder for it to become that nice crystal structure. So those will be the ones that'll stay liquid at room temperature. But it works the same way in when you're thinking about super, super cold, right? The more of this that you have, the unsaturated, the harder it's gonna be to build that crystal structure, aka you're not gonna freeze even in like the North South Pole. Good question. Any other questions?
So unsaturated is better with high temperatures?
Unsaturated. Unsaturated helps you not freeze at low temperatures. Unsaturated helps you at high temperatures. Let me see how long it takes to cook up. Let me see. Saturated, you're gonna have vertical flows. You're gonna put energy into it. So here, right, we said we have 19 carbons. When you have another molecule interacting with it, you have about half of them interacting with this thing, right? Like the van der Waals interaction. Every one of those van der Waals interactions, you have to break it to get that molecule to get released, right? So in this case, the more of these nicely arranged stuff that you have, you need more energy to break them apart from each other. On this side, you're gonna have this type of thing, right? So in which case, you're gonna have this interaction might be pretty similar to that first one over there, but this interaction is gonna be limited. Right? Because of that, that means when we get to the hotter temperatures, this is gonna make it easier to break this guy off of here. So this will help you when it's hotter to be more stable. This is probably gonna be a little bit less stable.
Is that what they call a free radical when it breaks off?
No, free radical, something else.
A free radical is when it's one like electron from a lone pair, right? That broke off.
So there would be, I'm thinking of like the whole molecule is leaving. The free radical is like if you start breaking bonds between them. Yeah, so, but normally when we're cooking, we're kind of sitting within this range what we're making. I can't remember how hot you have to get for free radicals, but you're gonna need more energy to make that happen. But if you get it hot enough, yes, you can make it happen. Any questions? No? All right, cool. See you guys on Wednesday.